Right, so I think we can start. Well, welcome to the last lecture of everything. Thank you for staying until the very end. So today we're gonna see the most important part of the lectures, with a lot of hard things to remember. So you have a very hard exam on Friday. Now we're gonna finish what we were seeing yesterday. We're gonna see the other types of new phenomena that we can find in when we have these partial differential equations. And then I think it's also a good idea to show you a more maybe a more realistic application to these models. We're gonna see, without going into a lot of detail, but kind of more or less the same math that we have been studying this week, how you can apply that to a two-dimensional system that has actually an ecological meaning, and what can we learn from that system, which I think is interesting to connect the whole thing we've been seeing the last days with maybe some more real-world example. And at the end, if we have some time, I'm also going to show you other different mechanisms by which we can get this type of solutions that we are going to see today. And we are going to go into them trying to develop intuition, because I don't think we will have time to do the whole derivation. Okay. So we start. So yesterday we saw how we have these traveling waves, these solutions that basically consist on a front that is propagating in a given direction. And today we are going to be studying solutions that are stationary in time, but have some regularities in space. And I'm going to call those solutions patterns, right? We're going to see very, very soon what I mean by patterns. So those are very, very interesting solutions of these PDEs. They are very interesting itself structures, because when we look around us in biological systems, we find them in many different places. So for instance, these three pictures that you can see now, the one on your left is the cornea of some insect. It's a very, very small scale. This is just a few nanometers. But also the eye itself of the flies also has some kind of regular structure, as you can see. And even, of course, in ourselves, we have a lot of patterns. This is just the fingerprint that we all have. And this, we can say it's in the scale of nanometers to centimeters, more or less. Looking around us, there are many, many other examples. I don't know if you already have some of them in mind. But scaling to larger systems, we have around us a lot of animals that have also very beautiful patterns. So this is a fish, two fishes, some ray, and then a leopard. They have these, their skins or their coats are also somehow, they show some regularities in the pigmentation. And even going to the very, very, very large systems, here we can say we are in the scales of centimeters to meters, more or less. And in the meters to kilometer scales, we have these patterns that also cover very, very, very large regions. This is vegetation, grass that is forming this kind of labyrinths. This is a landscape that is actually very beautiful. It's in Namibia, in Africa. And it basically consists on a very, very large desert with some vegetation, and these holes that are very regularly distributed within the vegetation. This is called the fiery circles. It's, if you Google that, you will find a lot of pictures. This is a coral reef where we see that there's this kind of rings as well. And this is a picture of mussels, how they arrange in the sea when they are growing. So the common thing of all these phenomena, you might think of many, many others, is that the way that these different components, the different things that compose this system are arranging in space is very regular, to some extent. Or at least this has some spatial distributions. And the idea that we want to approach today is how can we, as we've been doing these days, how can we construct mathematical models that give us this kind of that reproduce this kind of patterns, this kind of solutions, and what can we learn from those models? So we're going to work with the kind of equations we had yesterday, actually with the exact same equation to begin with. Whereas if you remember, we had I'm going to use rho today as a density instead of concentration. We had that the temporal evolution of that density was a function of some production plus some diffusion, right? And the idea is to see whether in the stationary state, so when the temporal evolution of my density, whether I have, which kind of solutions do I have, let's say, to be more general. So in principle, this equation will have a series of fixed points. If we think of the quadratic terms that we had yesterday, this has like this 0 and 1, if you remember. But in principle, we are going to have some rho star solutions for which f of rho star is equal to 0, right? And if we assume that these solutions are homogeneous in space, then this derivative is going to be 0, and therefore the homogeneous solutions of this equation are the same as the ones for this equation in the stationary state. Okay? And we know, we already learned during the week that if I introduce a perturbation to those solutions, I can see whether they are stable and unstable depending on whether the perturbation is going to grow or the perturbation is going to die. But the idea now is to ask whether if those perturbations, instead of being, as we were doing before, only time dependent, whether if when those perturbations also depend on space, whether those fixed points are going to be stable against those perturbations or whether they are going to be unstable. Okay? And the way of asking that question, of answering that question, what do you think is the way to try to study the stability of these equations against spatial perturbations? We've been doing it, maybe this is the 5th time we're going to do that with a small change. Well, I can study the linear stability of this equation against those perturbations, right? So now the only thing I have to do is to assume that my density rho is equal to the stationary solution plus some small perturbation that now is a function of space and time. Okay? Doing the same analysis we've been doing these days, if I basically repeat the same, I'm not going to repeat it. Right? This is 0, and this. Okay? So how can I solve this equation? Considering that I have this new contribution here. How many of you have seen Fourier analysis? Fourier? Okay, so then it's going to be faster than what I thought. That's good. I can transform this equation in Fourier, right? What's the Fourier transform of this? Okay. So basically what I have is right? And if I assume that my perturbation is proportional, my perturbation in time is proportional to some to some exponential, then this derivative is basically the growth rate times the Fourier transform. And then basically what I get at the end, I'm going to write it here, is okay. Any questions so far? Are we all on the same side? The only thing I've done is the same analysis I was doing yesterday, but because my perturbation depends on time, now of course my growth rate is going to depend on the modes, on the wave number of those of that perturbation. So in which conditions I will have patterns given this lambda? What do I need for my solution to be at least for some k unstable? Sorry? Lambda positive, right? That's right, right? At least for some k. And then I need, because d is positive, k squared is positive. If I want this to be positive, how can I say about this contribution? Sorry? Has to be positive, has to be negative, has to be positive, right? But what happens if this is positive? If this is positive, what happens with the solution? Sorry? No, if this is positive, how is my fixed point? Saddle point, I'm in one dimension. Why I can't? If I have a fixed point, and the derivative of the function is positive, evaluated at that fixed point, my fixed point is unstable, right? Because if I do this linear stability analysis with a perturbation that does not depend on space, that was the condition that I got the first day. So therefore, if my solution is stable against perturbations that do not depend on space, then this also has to be negative. And therefore, there is no way for this to be positive if in the conditions that I am studying so far. Have you all followed this reasoning? We can repeat it again. We are not.
I didn't understand why lambda has to be positive.
So the lambda is the growth rate of my perturbation. So if I want it to be unstable for some k, and therefore have some wave numbers that are unstable, then I need lambda k to be positive. And if lambda k has to be positive, given that all these numbers are positive, and this has a minus sign in front, then I need this to be positive. But if this is positive, my fixed point is unstable against perturbations that do not depend on space.
So this is implying that I can be stable in time and space at the same time. So my perturbation in time, if it is stable, in space I won't get something stable.
If your perturbation is unstable in time, then it's not stable in space. Yeah. Sorry, I had to repeat. So then.
But you can always have a k big enough so lambda will be negative, infinite case. So you always get infinite negative lambdas, right?
From k?
Yeah.
K is to the square.
Yeah. No, so you have something minus b k squared. I can always choose a k that's bigger than f prime over d.
Yeah.
So you will always have negative lambdas.
Yeah, you always have. But you will, I mean, so negative lambdas are stable. Okay? So what you are saying, which is totally right, is that when k goes to infinity, this will always do this. No? But the idea is whether this is always this or whether there are some case where this lambda is positive.
Okay.
Because I want to see when the perturbations, instead of dying, are amplified and when there is some spatial instability. And then I can have this kind of solutions. Does it make sense? So one of the one of the mechanisms that we are going to, well, the one that we're going to study more in depth that can give new possibilities is considering that that the interactions between our individuals that are leading this population dynamics, instead of happening locally, so instead of saying that this is a function of rho at the position where these individuals are, we are going to assume that if we are going to consider the case in which if we have some individual at a given position, this population dynamics depends on the population around within some long-range neighborhood. So if you remember yesterday when we were doing the derivation of the equation for the flux, we truncated that derivation at the first term. And we said that when you have like more dense populations, or in some conditions, you cannot do that truncation, because that gives you like a bad approximation. So what we are considering now with this case is cases in which that approximation is not correct, and then we consider somehow the whole range of interactions. Okay? So now, what we are going to have is an equation where if we assume that f of rho, just to keep things as they were, if this is equal to b, right? This is what we had the first day. You can rewrite it in many different ways. If you remember, at some point we wrote something like this. Then with k equal to 1, it doesn't matter. At the end, it's basically the same. So we are going to consider that this So we are going to consider that this function now accounts for what is called a non-local term in space. So now there is a question here. What's the meaning of this kind of solution, of this kind of choice, and why do you think it makes sense to consider it here? This comes back to the very, 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 very first class, the very, very first approximations that we were making.
So if the individuals are close, then they die far away?
Yeah, that's the idea. The idea is that now, instead of needing my individuals to actually somehow collide or find each other, like cross their trajectories, what I'm saying is that there is a neighborhood around each individual in which these interactions can potentially take place. That's the idea of choosing this term here, which is basically nothing else but an average density of individuals. And I'm going to keep things very, very simple. I'm going to assume that it doesn't matter how far every individual is from the focal position. Let's say that here is where I have the individual. It doesn't matter how far those individuals are. They have the same weight on the average. But maybe you are already thinking that if I choose different functions for this kernel, this is called a kernel, If I choose different functions for this kernel, what I'm doing is nothing else but modulating the intensity of the interactions with the distance from the individual. Okay? Does it make sense, all this?
Isn't there a prime in the x and the rule of xt? Otherwise, it gets out of control.
25 minutes, not that bad today. So now the only thing I have to do is to repeat the same analysis that I did before, but considering that my f of rho is now this non-local function. That's it. I can assume that rho is, again, some stationary state plus some perturbation and linearize and do everything. I'm just going to copy what you get, that hopefully in the notes is correct. So this is an intermediate step that you get before transforming in Fourier. When you transform in Fourier, how is this term looking like? This is a convolution, right? And the Fourier transform of a convolution is the product of the Fourier transform. Okay, so that was just a brief parenthesis, so you don't get lost with the result. And at the end, what I get for, of course, I have to consider also that there is this rho 0. What's the value of this rho 0? Sorry. What's the value of rho star? Yeah. Which one? You are pointing to it. I'm assuming that rho star is a non-homogeneous solution in space. So therefore, this, I mean, if I assume that rho is not a function of a space, this is the same as this, right? And the fixed points are and the other one was B over mu, huh? Yeah. So rho star is B over mu. Okay? 0, I know. I mean, I'm interested on the path on the solutions around the fixed point that has population. Well, and then at the end, we get growth rate for a perturbation that looks like this. This is the one that we're going to look at now. And to have patterns, this, we said, has to be 0 at least for some k, some critical k. Can I make this to be positive for some k, or I can't? Sorry? I need g of k to be negative for some k. So now we can come back here to this choice we made for the kernel function. I don't know if, well, do you know what's the Fourier transform of this function? No, well, it's the sine. Okay, right? And this looks something like this. There should be some, it is. There should be some observation here. So this has some, this is negative for some wave numbers. So in principle, I could have patterns here, right? I could have this to be positive for some patterns. And just so you know, this R here, so basically the width of this kernel here is going to give me the frequency of these oscillations, right? So in principle, the actual K at which this lambda becomes positive is going to depend, of course, on the R that I'm fixing the kernel. So depending on the range of my interactions, I will have a characteristic scale in my pattern. This is basically what this is telling us. And of course, we can calculate the derivative of this lambda and try to find the maxima and then see the conditions. The only thing is that, of course, I mean, when you calculate the derivative of this, you lose. I mean, you cannot calculate this like analytical. But we can gain some intuition about the biological conditions. So what does this mean in terms of the biology of the system, right? Which at the end is what I'm trying to understand. How would you try to gain some intuition on this? For example, there is some idea. I'm going to clean this up while you think a little bit. So in every case we've studied these 4 days, whenever we were not able to do some analytical calculation, we've done the same thing. Yeah, good. Sorry? Well, we can try to see if we learn something by drawing this function, no? So if we can rewrite this as a non-dimensional growth rate by dividing by b, and then this is. So which kind of plot do you think it would be informative here? What's the contribution of this term here? Well, it's a parabola, right? But I mean, from which kind of, from which mechanism, from which process is this coming? I have two processes in this equation, right? I have movement and I have reproduction. This is coming from the movement term, right? The diffusion. And this is coming from the interactions in the reproduction. So in principle, I can write something like this, which will be kind of this, no? And I have my kernel. This is going to be a challenge. Well, not too bad. So what's happening here in this case that I've drawn here? There is no pattern formation. Why? Right. We all see that, right? But then if I play with this ratio, I'm changing the slope of the parabola, right? Well, you all see what I mean, right? And there will be a value at which I actually, each curve touches each other, and then that's the critical point at which I get patterns. So basically, by this very simple sketch, what I have is that depending on the ratio between the diffusivity in the system, so depending on how fast my individuals are moving around or how well they are mixing with each other, diffusion, we said yesterday that it was mixing things, and depending on how they reproduce, we may get these patterns or not. And of course, you can always plot the whole thing, which is what you saw yesterday like 5 or 6 times. So this is this is basically this lambda k as a function of k for 3 different values of b. So when b is 0.4, as you see, that's the black line, the one in the bottom. And lambda is negative for every single k. I never cross the 0 line. As I increase b, at some point, I do have some wave numbers that become unstable, and I can have, in principle, according to our analysis, I can have patterns, right? And then as they increase, they go. So the other thing that we can do is to actually check whether, I mean, this is giving us a kind of a guide, right? I know that for these parameters, I should expect having patterns. So that saves a lot of time when, for instance, we want to actually solve these equations numerically to see how the patterns are. This is guiding us. It's giving us a target that we can attack directly instead of like doing a blind search. But we can do that integration. There are numerical methods to integrate these partial differential equations. I don't know if any of you is familiar with any of them. But the thing that I'm going to show you is just the integration of this equation, well, with this non-locality, for the set of parameters that you have up there, okay? What I'm doing is starting from a density 8, y8. Because, well, A is mu. So basically, 8 is my fixed point, my homogeneous solution. And the reason why you see that this is like a thicker line is just because I'm adding that perturbation. So I'm generating some random number, and I'm adding it up to the homogeneous solution. And now I'm going to integrate the equation on time and space to see what happens. Sorry, what should happen? What do you think is going to happen? Still half of the movie, so anything can happen. What would you expect? I would expect not getting patterns, right? Especially because we saw before that B 0.4 doesn't give patterns. So the only thing that the system is doing is to amortiguate, to make the perturbation to decay, and then I recover the homogeneous state. But now if we increase a little bit B, so now it's 0. No, it's the same slide. Now it's not. If I have B 0.5, then the story should change. I should have some wave numbers that are unstable, and I should get patterns. So let's see what happens. So now, because I do have those, I mean, I have this point to be unstable like for some wave numbers, what I get is a solution that has this oscillation. Do you think we can know something about the wavelength of this solution, of this pattern? So this is a periodic solution, right? Can I know something about the periodicity, or I can't? It's a tricky question, a little bit. Good question for the last lecture. So in principle, I know that I have only a range of wave numbers that are unstable, right? And actually, the maximum should be the first one becoming unstable. So the wavelength of my solution, of my pattern, should be on the range of the wave numbers that are unstable, because the other ones are decaying, right? So here, well, it's hard to say, but the periodicity of the pattern, which is of the order of 15, is related to 1 over 0. So it's related to the wave numbers that are unstable. Okay. So this is so far what I wanted you to know about the possible solutions that you can get from these kind of partial differential equations and the kind of mechanisms that can lead to these patterns. So the first thing is that I do need, I mean, with these mechanisms that we were considering yesterday, there is no, I mean, in principle, I don't have patterns. I need for additional things. One of them is these non-localities that we introduced, but there are many others. What I want you to show now, like for 15 minutes, 20 minutes, it's what kind of, I mean, how can we use all these tools that don't seem to be, I mean, very sophisticated to try to understand a real world problem, right? So this is probably going to be less tiring for you, hopefully. So we were saying that one of the examples where we have these patterns are these landscapes of vegetation, right? So these are satellite images of some regions of the Earth where we have the vegetation forming kind of regular structures. In the first case, we have the soil with some spots of some plants that are interspersed on the soil. In the other one, we have kind of more stripes or labyrinths, something like like a tiger, the coat of a tiger. And in the other one, what we have is almost everything green with only some holes, similar to the fairy circles that we were seeing yesterday, before. Similar. I mean, and there is something that is very interesting about these patterns that you are seeing here, is that they appear in many, many different places. Although you may think they don't, they have been found in Australia, in Africa, and they usually appear in regions where there is not a lot of water. So when there is some aridity and plants need to fight with each other to get access to that water, then they tend to organize in these shapes. And there is something additional. I mean, there is something that makes these patterns even more interesting, and it is that now, because we have many different ways to look into the Earth from above, we can use satellites, for instance. We all can do it actually with Google Maps. We can identify these patterns and we can analyze them. And this is something that some people did. And actually, what they found, they went to some, they studied a region in Africa. They took a lot of pictures from the vegetation in that region, and they analyzed the type of patterns that they were finding. And they classified those patterns into the 3 categories that I was showing you before. When it says labyrinth in yellow and bandit in red, it's kind of the same. It's kind of the tigerish pattern that we were saying before, right? The only difference is that sometimes because, well, you have some slope, the pattern doesn't look the same. But for us, they are going to be exactly the same thing. So basically, they put a dot of a different color depending on the type of pattern that they found, and they try to correlate that with the amount of water that they have. And they found, as you can see, every dashed line marks an amount of water that region has. And they found that as water becomes more scarce, so from 550 to 500 to wherever, the shapes of the pattern was correlated with that gradient. So when there is more water, we have a bunch of green dots. As the ecosystem becomes more arid, then we transition to red. And then at the end, we transition to yellow and finally to blue. So there is some correlation between how these patterns look like and the amount of water that the system has, the amount of precipitation.
But the vegetation is the same in different colors, but it's different vegetation.
So there is different vegetations, but the thing is that the vegetation,
Then maybe no correlation, it's just different vegetation from different patterns.
No, because there is not exactly the same vegetation, but there is, so you have, the thing is that whenever you look for these patterns, you'll find this gradient, regardless of and you have only these 3 patterns, regardless of the type of vegetation that you have. So of course, yeah, it could be, you may have different plants that have a tendency to form different patterns, and probably that's why the transition is not perfect, but the idea is to try to see if there is something more general hidden behind. So the idea is precisely whether there is something hidden behind this correlation, or if it's something that doesn't mean anything, or if it is just because there are different types of vegetation forming the patterns. And to do that, what people have been trying to do is to develop these kind of models that we've been seeing before using PDEs. And actually, the one that I'm going to show you today is very, very easy, very simple, and I'm going to show you the kind of results that one can reach at the end. So the only assumption that this model does is to assume that the main interaction between plants is fighting for water, and that competition for water is mediated by the roots, which is, I think, an assumption almost in which we will almost agree as much as in the one we made the first day, that living things die and reproduce, die and move. Its principle sounds reasonable. So we'll see. Let's see how far we can get with that. So let's try to write. Let's try to write our model. We're going to assume that the first thing that happens is that the vegetation is dying at a constant rate. As our particles were reproducing at a constant rate the first day, now they will die, which will lead to a term like this, right? As we had the one here, so now it's the same, but instead of adding particles, we are removing them. And now we're going to assume that plants reproduce in 3 steps. I'm going to draw you the 3 steps. You see how easy this is. The first step is that whenever we have a plant, it produces a seed. The seed is going to fall very close from the plant that produced the seed. And then, depending on whether, depending on how many plants are around that position, then that seed may germinate and produce a new plant, or it may not, right? So if we have that the production of seeds is constant, as we assume the first day, we have a term like this. But now we need to account for the fact that this space is limited, right? So if a seed lands here and everything is already occupied, then there is no way for it to land. So if I assume that rho goes between 0 and 1, then basically this is what this is telling me, that the contribution to the birth, if rho is equal to 1, is 0. And then I'm saying that I need a probability of establishment that I'm going to call PC, which is the probability of overcoming that competition. So we have something like this. The question is why this probability depends on the rho tilde is the non-local density as we had here. So why that probability depends on that instead of on the local density? So one of the assumptions that we made is that the competition for water, in this case, is mediated by the roots. And we said that the roots have a longer range than the plant itself. And that's the idea of this kind of non-local terms, whenever we have them in this kind of toy models, is to account for processes that are driven by interactions that do not require the encounter of these two particles, but they can be far away from each other. And we know, I mean, even if we want to keep things general, because we have no information about this PC, I mean, what's this PC? Is an exponential? I don't know. But I know something, right? I know some properties. I know that the more plants I have around me, the less likely I am to germinate, because the competition is stronger. So I know that this derivative has to be equal to 0. And I know that when I can assume that when there is no plants around, delta is giving us the intensity of the competition, so it's the amount of water that I have. So when the competition is equal to 0, when this intensity of the fight is equal to 0, then my probability is equal to 1, and when this intensity is very, very strong, then this probability is equal to 0. Okay? So at least I know some general properties about this function that will help me in my analysis up to some point. Then, of course, I always, in any model that we make, we have to make choices, and we have to make finer assumptions, and then we need to see how robust our results are against these choices.
What is this delta?
So delta, my PC, my probability of overcoming the competition, is a function of how many trees there are around that position, and this parameter delta that it's giving us the intensity of water, the amount of water in the system, so the amount of the intensity of the fight for water. So just to give you a more concrete example, if I have this PC to be an exponential with the amount of vegetation, my exponential will look something like this, right? So delta modulates the decay of this function. It's like a modulating parameter. I want to stop here to tell you that, of course, as you probably are imagining, this is not, I mean, you don't find anything like this in the exam if you are worried at this point about this. I just want to give you some example, okay? And I can do the whole linear stability analysis that we've been doing in those more simple equations, and I can get this lambda of k that is giving me some intuition about when I could find these solutions and when I should not. Yes? This is the non-local term without any of this. Huh? It's just the average density. And we have this lambda of k, and in principle, we can do the same as we did before. Of course, I need to make some choices for these functions, and then I have to test whether my results are depend on those choices or not, but in principle, I have some guiding. Well, we can make these choices just because this makes the equation more easy to solve. And I can plot my lambda k, and I can do exactly the same thing. And I can see how, as I regulate the amount of water, remember that this delta was the amount of water, so how I get a transition from not having patterns to having patterns as I regulate the amount of water. And something that is already interesting is that as I reduce the amount of water, remember that delta is the intensity of the competition, so the larger delta is, the less water I have. As I reduce the amount of water, I find these patterns, because I find this lambda k becoming positive for some value. I can do many other things, but just because I don't want to tire you a lot, I can solve this equation for different values of lambda, and I can see how these patterns look like. And I'm doing this in 2 dimensions because the Earth, I mean, the landscapes, the landscapes are 2-dimensional, the ones that I understand, the surface. So what I find is that as I decrease the amount of water, I get this transition from having vegetation everywhere to then having these holes of soil, to then having these labyrinths, to then these spots, and then I lose all the vegetation. So in principle, with this there are many other things that one can do and many other analyses that I don't want to go into these details with you. The only thing I want to show you is how this very, very simple model with very few assumptions reproduces qualitatively the behavior, the trend in these patterns that we observe in these images. Okay, that's the...
These figures are the numeric simulation
yes, from that equation. And the choice that I'm making for G is basically the extension to 2 dimensions of this kernel. Okay. Well, anyway, the thing now is so here it's a little bit harder to see, but we have the same condition. We have that in order to have this lambda k to be positive, G of k needs to be negative at some point. And that appears in many, many different models that use that make this series of assumptions and use these non-local terms. And the idea is, what is the biological mechanism that this model is encoding that gives these patterns? And I'm just going to give you the idea in case you are curious. What is happening is that when we have our plants growing in different places in the space, at some point, whenever this is done, we will have a plant that is going to try to grow in a place where it's surrounded by a lot of plants. Okay? And this R is basically the range of the interaction that this plant has with the neighbors. And let's say that it's successful there, and we have a new tree, and we are happy. But then we may have plants trying to appear in a place where, because of the range of the interactions, they not only compete with the ones in their clump, but with plants in both in both clamps, and then the competition is stronger, and in these regions, plants disappear, and they reshape the distribution, the spatial distribution of plants, leading to this formation of these regularities. And as I make the probability of surviving smaller, then at the end, I move from shaping these exclusion areas that I'm calling here to shape them from being these holes to being almost the entire landscape. That's the idea behind this, just very briefly. And just to finish, and before we go to questions, so I told you in the beginning that this is only one way in which you can get patterns, and actually it's not the most frequent. It's not the one that people more usually rely on, but it's the one that, at least I thought, has the most simple mathematical analysis to get an idea of what's happening here. The other mechanism that people, that is much more frequently invoked to explain these patterns is what is called the, it's based on two reaction diffusion equations as the ones that we have here, and it was proposed by Alan Turing in the 50s, okay? I'm just going to give you, we're not going to do the mathematical analysis, the whole thing. I'm going to show you the model that he proposed, the intuition behind the formation of the pattern in that model, and then we are going to try to think a little bit to see if from there we've developed intuition enough to find the conditions, the mathematical conditions that we would impose to that system, okay? This is an exercise just to think a little bit and for you to know an alternative mechanism to form this pattern.
I have a question.
Yes.
Can you explain why is there an exclusion error? I didn't get it.
So the idea is when you have a lot of plants here, if you want to think in terms of plants instead of in terms of the equations, you have the roots of this plant reach up to here, right? If I have another clamp here, the roots reach up to here. So the idea of those exclusion areas is that in this area here, you have roots from this patch and from this patch. So you have, let's say, twice as much roots as only here. That's the intuition.
So another plant can't go find this root space.
Yes, that's the idea. No plant can invade those root space, and the ones that by chance were in the beginning, if you say that you start like throwing plants randomly, they will eventually die because the birth is, the reproduction term is 0, and they all have some probability of dying. That's the idea. Of course, yeah. No.
In a more realistic model, I think that maybe if you're closer to the other plant, maybe the competition is kind of harder than if you're more far away. I don't know, because the roots are more strong close to the plant.
And how?
You change the G.
So that's the idea, of course. And this is something I want to say, like a disclaimer. The model that I presented you, it's like super simplified. It's one of the possibilities that we propose, but there are like a lot of things proposed. Again, I chose that one because it was very close to the equations that we've seen the last days, and I thought it was going to be easier for you to follow the whole thing. But of course, it has a lot of weakness, and one of them is that we are assuming this G, whereas something like this would be way more realistic. In the neighborhood of the plant, the competition is is more strong, and as I move away, the competition is weaker. There are many other things that this model doesn't have. So, for instance, and many people do that whenever there is a plant very, very close to the plant, usually there is a facilitation mechanism that makes this region better for plants to establish, because the fact that there are plants reduces how water erosion in the soil, and leaves that fall make that soil richer, and people also try to introduce that, and there are many other simplifications. But the one that you were pointing out is one of them, and the way of solving it is with changing the G.
And what about the boundary conditions for this equation?
So in the plots that you saw are periodic boundary conditions, which means that the neighbor of this position is this one. Well, the neighbor on the side. The neighbor of this one is this one, and so on and so forth. Okay, so that's a way of minimizing the effect of having a small system. Okay, it's kind of simulating a snapshot, like a zoom on a region that is much larger. It's just you simulate the IC model, for instance, or.
So besides predicting the pattern formation, can you also predict the length, the characteristic length that appeared in nature?
So in nature, what we can do, we can measure the characteristic length of the real pattern. If you do a Fourier transform to the image that you have at the end, you can get like the characteristic wavelengths. And in your models, the wavelength of the pattern is related to this maximum here. And something that one can do, because we know that this is related to the kernel that we choose, is to try to see whether the conditions that you need in the model, I mean, you can choose your kernel to fit the wavelength that you observe in nature, for instance.
You cannot predict in advance what the K would be by like studying how the birth and death processes of those.
Well, what you can do is if you have a picture that you know is by some species, you know the species of plants that are living there, and you know the size of the roots, then you can see whether having that size of roots in your model gives you the pattern of the wavelength of the pattern that you observe in nature. You know what I mean? Because we're saying that this J was related to the extension of the roots, right? So in principle, if I know that this plant has, I don't know, like 20 centimeters, then I can try to see whether my real pattern has the wavelength that I should expect from that kernel, for instance. That's a prediction of. Sorry, I thought there was another question. So the very last thing. Let's see, one dimension. So what Turing suggested was he introduced a system of two reaction diffusion equations that are coupled with each other, and each of them has different diffusivities. This kind of system, the way it produces patterns, I'm going to give you the mechanism, right? Okay, with words, and we're going to draw it. He suggested this is an activator, and this is something that we'll call the inhibitor. And the idea is that whenever we have both of them together in some place, the activator U activates both itself and the production of the inhibitor. Whereas the inhibitor agrees with its name and inhibits the activator. And they both diffuse. Okay? So if we have, let's say, two profiles that are somehow random of both things, this is not very random. So they say that this is V and this is U. the idea is that whenever we have a profile like this, because U activates itself and also activates V, in these places we have the production of both the activator and the inhibitor. And then what's the idea? The idea is that these two things, these two species, or these two chemicals, they both diffuse. And the condition that he found for having the patterns, and this is the condition for having patterns in this kind of system, is that the diffusion of the inhibitor has to be way faster than the diffusion of the activator. The question is why? Why? So the idea is that because the inhibitor diffuses a lot, it leaves, right? And then the activator can grow, and the inhibitor, whenever it leaves, it inhibits the activator. And at some point over time, I get amplification of the activator here and amplification of the inhibitor in there. Of course, this is because we had 10 minutes, but you can obtain the, and that's actually what he did in his paper. It's a paper from 1952, I think. You can calculate the conditions. You can calculate this condition given that system and for those functions. The way to calculate that is basically doing linear stability analysis as we've been doing before with the information that we already do have. We have some information already about this, right? Or we don't? We do. Do we know something about this function? Because we have this, right? So we know that the production of U increases when I have more U, and so on and so forth. So when I calculate the Jacobian of my linear stability analysis, because this is a two-dimensional system, I do know the signs of the derivatives. And from there, you can elaborate, and you can obtain at the end that this is required to have patterns, basically, in this kind of system. So he was working this system with the idea of understanding morphogenesis, which is how systems how organisms form. But these kind of models have been applied to very different systems. One is the vegetation patterns that I was showing you before, where the activator is the water, the inhibitor is the water, and the activator is the vegetation. And the reason for that is because the lack of one activates, the lack of one inhibits the other. Is an argument that is a little bit hard to follow, but of course water has to diffuse much faster than vegetation. Another system is the fishes that I was showing you before. These patterns in the fishes, in the leopard, and in many other systems. And actually, if you Google these Turing patterns, there are a lot of outreach articles, or like written by scientists, but not very technical, where they explain all the different applications that they are trying to work with this. And this is a way more frequent mechanism to invoke the formation of these patterns. Again, I thought that the linear stability analysis was easier to follow in one dimension, plus you also learn how to deal with these non-localities, and that's why we work with the other one. Okay, but this, when you go and find different pattern formation models, this is much more. There are many more works trying to explain patterns with this. Okay. So now questions, comments, complaints?
So if there's no local equation and this couple of equations have the same effect of pattern formation, could you go from one to another directly, like removing the influence of V but in a no local influence on the first equation directly, like that.
I tried to do that for 2 years on my PhD. So that's, it took me also 1 year to make myself that question. So the idea is whether if these two formulations that I was showing you before, if they give the same kind of structures, whether I can go from one to the other. No? If I can reduce this system of two into one, and then I don't lose the information. No, that's the question. So I could do it. The only thing is that so when you do the calculation at the end, when you remove this variable, that is the one that you don't want to look at, let's say, inhibitor, you encode that information in the function g. And I don't know if, well, I mean, you do an adiabatic, what I did, that for sure was not the right way to do it. You do an adiabatic elimination. I don't know if you know what does it. It means that because one of the variables diffuses much faster than the other, you can get rid of this somehow. And at the end, the thing is that the g function that you get doesn't meet the conditions of for some k. So the j's I got, the g's I got, are always positive for any k. So the question is there, if you know how to answer it. Yeah.
You did a simulation for the spatial structure and it showed a oscillating pattern. Does the amplitude converge?
What do you mean converge? If it yeah. At some point you get the, especially, and indeed, for instance, if you have one of the exercises that you have in the web page, it's about doing this kind of analysis, but in an equation where you have the non-localities in the diffusion instead of in the reaction. And in that case, because we get rid of this production of individuals, the rho is conserved. And when you do the simulation, if you are doing it right, and you integrate over the whole space, rho should be constant because it's conserved. And at the end, what you get is that what you are putting in one side, you are removing it from the other, basically.
Does this model only reproduce the patterns in the animals, or do they explain, I don't know, like in terms of chemicals in the skin of them? I don't know, like why do they form? Like why? Like how they became like this? Not only reproduce like how they are.
Well, the idea is this: the way the two chemicals interact with each other is in these functions. So when you get the conditions for these functions to give you the patterns, you kind of get the kind of interaction that you have. And what also predicts is it predicts usually the shape as you play with this. Once you know these functions, you have one parameter that is going to be your what is called a control parameter. That basically, as you move that parameter, you change the shape of the pattern change basically, and you can play with. As you can see there, you have like the same spots, stripes, and I didn't put any labyrinth. But this, what is characteristic about these equations it depends a lot on the local term. But for the local terms that he was proposing, the production terms he was proposing, you get these three things. You get spots, you get labyrinths, and you get spots, labyrinths, and gaps. If you change this and you put like more complicated functions, you may get like spatiotemporal patterns, like spiral waves and things like that.
He was predicting the existence of two chemicals, is that right?
No, he knew that there were two chemicals. He was predicting.
No, he knew for which of these patterns there were two chemicals. I'm trying to think, how do you check if this is really what's going on? So the claim is that there really is two chemicals that make these patterns.
No, the claim is that when you have two chemicals, you may have a system that is stable, and it becomes stable for differences in the diffusion. So that diffusion.
Sorry, when you say chemical, you really mean chemical, right?
Yeah. In the original.
So for plants, this is irrelevant. This is not vegetative. This is some kind of fish. Is that the thing you're saying?
Well, when you move, so the thing is that the same the same kind of structure, mathematical models, have been used for the skins and also for the plants.
I agree you can build mathematical models for anything, but I'm trying to think if the mathematical model corresponds to physics or not, or it's just a model. So for this to be physics, you really need a mu and a nu. It's not just a...
Yes, sure. So in the case of vegetation and water, U is the plants and V is the water.
Why U a plant and U a water?
Well, because you write. So basically what you do is you write equations for your two species, and then in this F and this G, you code the processes that you want to look at. So in the case of plants and vegetation, here you encode water intake, conversion from water to vegetation with functions that I don't know by heart, but for instance, in vegetation, you encode evaporation of water for water. You also have the water intake, which in this case is a loss of water. So basically, the terms of each of these, the contributions for these two functions are chosen depending on the mechanism, on the system that you want to study. The idea is that the instability that leads you to these patterns is driven by differences in this diffusivity, plus nonlinearities here, of course.
Patterns are formed by just two competing things. Is that the idea? Is that the basic idea of Turing?
No, they have, well, the idea of Turing is that they compete. There is an activation. Sorry, there is an inhibition. They compete, but there is also an activation. So the idea of Turing is the activation and inhibition of two species. With two species, he calls it a species, like two elements with differences in the diffusivity leads to this pattern. So for instance, in our skin, what gives us the color of the skin is our molecules, is some, right? So in the case of the leopard, when they are developing from the embryo to the adult individual, when the individual is developing, then there are these two chemicals that are reacting.
So it's not genetic. They don't have, if you cut off a piece of skin, will it grow back the same or not?
Yeah.
It will grow back the same?
If you, this is hair. So basically, if you shave the animal, it will go back the same because what is giving you the color of the hair.
That's the genes that are already there.
Yeah.
The cells that are already there.
Yeah, but that's something, actually, like a few months ago, there was a talk of someone that is doing that. I was trying to see, not with leopards, with small rats, I think, what is driving the pigmentation of the animal. So he's trying to knock out, to play with the genes of the animal to see when, at which point when you shave the animal, the pigmentation changes. So I don't think, at least to me, that I don't know the answer. I don't know if you take a knife and you cut a piece of everything, I don't know how it will happen.
The claim is that the biochemists can find these two chemicals. That's what I'm trying to
Yeah.
So they can really find these two
Yeah.
chemicals?
More questions?
If you take into account more complicated internal dynamics of these populations or systems, because, for instance, we're considering the birth rate to be time independent. In some cases, that doesn't happen, right? There are seasons in which animals reproduce and then they don't in others. Does that change the overall solutions
Yeah, so it does, and it changes. It changes things in a way that depends on the way that you, of course, change your parameters. What I can tell you as an example, for example, if you, one of the things that I know people try to do at some point is in these models for vegetation, because we are saying that the amount of water is giving you this transition in the shape of the patterns. So what they were trying to do is once I have one pattern, play with this parameter with the reproduction parameter to decrease it to see what happens, no? To see if I see the transition of in the shape of the pattern from one shape to the other, or if what I see is that everyone dies, or if it's, you know what I mean? And it depends a lot on, for instance, on so if you reduce that birth rate, depending on the velocity at which you reduce that parameter as compared to the time scale that the system has, so depending on how fast your system reacts to that change. So basically there is a competition between how fast you change your parameter and how fast the system reacts to that change. So depending on how you play with those two time scales, you get either that the pattern changes, or you get that you basically have, let's say, those logarithms, and you reduce B a lot very fast, and basically where there were, in the places where there was vegetation, the whole vegetation dies. If you have your B oscillates, then sometimes you get your pattern going up and down, and if it is noise, you may get very different things. Like if your B changes with, I don't know, some random function of the time, you can get like very different things depending also on the way in the way which you are changing the parameter. More questions about anything?
Give an argument why you always go. You said you have three different types of patterns, right? And you have why do you always have to have the labyrinth? Is it one of the patterns? Why can't you go directly from one to the other without going through labyrinth?
Well, sometimes you do. You do go from. Actually, sometimes you go from the spots to losing the pattern. Depending on in other models, I'm saying. In this one, you mean in the equations or in the?
You say you have spots or you have gaps, and then in between you have labyrinths.
Yeah, for this particular. But you mean in the natural systems or in the models?
That's the models for the actual system. So.
Yeah, but the thing is that I mean, there are also natural systems in which you only have spots.
Yeah, sure. But suppose you have some variability like your plant, so that it goes from one to the other. You claim there's. It seems. You claim that there's always a region in which it goes through the labyrinth. Is that right?
Yeah, but that region, I always found at least a very, very tiny region where those patterns are a labyrinth.
It's just not clear to me why. Is there some mathematical reason why it always goes? Why can't the spots just get bigger until you just have gaps?
So I think
you have to go through the labyrinth.
If you do that's something I haven't done very, very, very frequently. If you go beyond the linear stability analysis and you do it's called a weekly nonlinear stability analysis to that PDE, you can get the geometry of the wave numbers that you can basically get the geometry of this pattern. So the spots that I was showing are hexagons. No? If you look into the lattice, they are hexagons, right?
Yeah, I see it.
So
why you have to go through that stage.
So the idea is, I think, when you do a nonlinear stability analysis, as you play with the parameter and you are looking into the angles that the wave numbers form, the transition forces you to have at least.
So I guess what I'm asking, is this a prediction of the model, or is this something that's just known about mathematical? So it's some.
No, it's known that these type of equations have this series of patterns. At least for.
To me, it's surprising. So it sounds like a prediction. I just don't know if this is something generic about the pattern. It's not clear why you just couldn't have the yellow spots just get bigger and bigger until it forms green. You understand what I'm asking?
Yeah.
It's a little bit surprising to me. Is this something just in 2 dimensions? Is it something that...
Well, I haven't integrated this in more than 2 dimensions. Because usually I'm interested in surfaces. So.
I understand, but you can ask the question in 3 dimensions too.
Yeah.
Sorry, but you said these were simulations from this equation, right?
Mm-hmm. No, from the one that was there before.
That one before, but this picture are 2-dimensional. So what are those dimensions? Isn't that a surface?
Yeah, but it was that equation in 2 dimensions. So x was x and y.
Equation, Laplacian two-dimensional.
But yeah, instead of being a derivative in x, it was a Laplacian. Yeah. Well, not sorry, it wasn't not. So, no, sorry, it was not this equation. It was the one that was, I mean, this one without Laplacian. Right. The only thing is that G is not like a hat function. It is a disk, and the Fourier transform is not a sinc function, it's a Bessel function. So it's in 2 dimensions, but this equation. And the brown and the green is a gradient between 0 and 1 in rho. It's not that it's nothing like this. More questions? No? Thank you.